and I'm trying different methods in order to try to understand what it is we mean when we speak about life. And so my title for today, if my slides will work, yes, is the development of lifelike droplets to study the fundamental properties of living systems. So really trying to study something fundamental about biology, but using a different kind of approach. I'm going to speak a little bit about, for example, natural living cells, but mostly I'm going to speak about chemistry and physics. And this talk is meant really for a general scientific audience. So slides will have some technical details, and I will not be presenting the technical details. I'll just be sort of presenting what is the thinking behind this and what are the lessons we're learning from these kinds of experiments. So I hope everyone can follow. Of course, you could also ask questions. So to start, I'll play a bunch of movies today. And here's the first one that was recorded by one of my PhD students. And you're just looking at a real-time image through a microscope of some interesting phenomena that's happening. That's one movie. Here's another one. So both of these movies are of non-living systems. They're just chemical droplets, which form the basis of my talk and the basis of my research for today. But I think you can gather already just by observing the phenomenology of these types of experiments that some interesting things might be going on. And you might think, if I didn't tell you, that this, for example, this movie here has some sort of group dynamics of living cells or organisms that are moving around and congregating together. But they're not. What they are simple chemical experiments, and they're based on this historical example by a zoologist named Büchli, who was studying protozoans like the amoeba. And he was interested in the actual dynamics and the physicality of these types of organisms. And so what he did was made droplets, and he activated them. And I think this is the first historical example of someone trying to make, let's say, an artificial organism for the purpose of science. And so what he did was made a dish. You're looking down at a dish that's a few centimeters in diameter here, with fresh olive oil in it. And what he added to it was water mixed with potash. And potash are the remnants of what you have when you burn wood. And it's high in potassium, so that's where the name potassium comes from potash, and particularly potassium hydroxide, which is a strong base. And so we took his recipes and redid them in the lab. And so here's a video of this. So first, I'm going to add a droplet of water with some red food coloring. Just stop this here. And this is just a droplet of water, red food coloring with no chemistry. So there's no chemical potential. This is just a control experiment. And then the next one I add is a blue droplet that has this potash or this hydroxide in it. And you can see very different behavior from this experiment. The one that has the chemical potential, the blue one, is dynamic. You can see the droplet is spreading through the olive oil. You'll see now it starts to break up into smaller droplets, and those smaller droplets start to move through the experiment. And so this is just an example of how you can take something that's inanimate and animate it through chemistry. In this case, it's very simple chemistry. It's just adding hydroxide to oil, in this case, olive oil, which is for you chemists out there, this is a saponification reaction. This is a reaction that you would use to make soap. So it's a very common chemical system that people have used for a very long time. And it produces these dynamic entities. And those, if you look at those with a microscope is where you get to these kinds of images here. Okay, so that's it. You take something that's like a droplet that doesn't do anything, you add some chemical potential, and then if the conditions are right, you start to then animate the droplet, and then you can observe what are they actually doing. And so what we can see from just that small set of experiments, some of the lifelike fundamental properties that we could sort of distill from those experiments. It seems like life as we know it needs some sort of embodiment or a body, which would be the droplet itself. It needs a source of energy if it's going to do anything. In this case, there's some sort of chemical potential, which is then used as energy. And it needs a means to transform things. So for example, transform energy or make and break chemical bonds in this case, which we might want to consider to be some sort of metabolism. It's not necessarily a biological metabolism, but it is making and breaking of bonds and harvesting some of that energy for, in this case, for example, having these droplets moving around in the system. So just from that small set of experiments, we start to then assemble a list of what are these lifelike properties we could study with these very simple chemical systems, again, that are not alive. In the previous example, it was just olive oil and some hydroxide. Okay, so we took this a bit further, and I'm going to talk about this dynamic droplet system too. And what this one is kind of, if you can see, can you guys see my pointer? Okay, great. So this is a illustration of a dish here, and we're going to put some water in here. We're going to put a droplet in there. In this case, so the outside, the environment is water, and the droplet is made of oil. In this case, nitrobenzene oil. So it has a density higher than 1. What that means is the droplet sinks into the water phase. Okay, and when you put nitrobenzene and water together, nothing happens. It's only when you then activate it with chemical potential. We'll use some fuel here called oleic anhydride, which is not so important. The important part is that this droplet is now full of this chemical potential. And then we can look down on that experiment and we can see what happens. So again, a droplet of oil that wouldn't do anything is now motile and moving through the system. So we've animated something. The chemical details are all on this slide. Again, I'm not going to go into those, but for those who are interested, you can see that's a complete list of what's happening here. And it's very simple, right? You're using a few simple chemicals, a few simple chemical conditions to run this kind of experiment where you can now have an animated droplet. And you could also see there's a trail behind that droplet, right? That whitish trail. That is because it's changing the pH locally. When this thing starts moving, when it starts burning the fuel that's on board, it's producing acids, and it's then using that as part of the mechanism for which it moves. What is important to understand, I guess, from this is if this droplet, using its onboard fuel, produces a pH gradient, which it appears to be doing, then perhaps what we can do as experimentalists is go into the system and add a pH gradient to the system and see how a droplet would respond. So that's this next set of experiments. I'll go back to that slide in a second. So here's that droplet moving around again. And now I will create an external pH gradient and see how the droplet will react to this. Okay, so the droplet's moving around kind of on its own, let's say. I don't know if it's autonomous or not. I wouldn't go as far as saying autonomous, but sort of independent movement. And then when I pattern the environment by adding a pH gradient, then the droplet seems to pause and reconfigure itself and then climbs to the highest part of that gradient. So basically we're able to control some of the behavior, control the motion, and show that we're basically dealing with a system of chemotaxis, so movement within different kind of external gradients, chemical gradients in this case. And the fuel is this. if you want to call this metabolism, I'm not sure, it's a bit of a stretch, but basically the fuel is this anhydride molecule that in the presence of water and high pH breaks down into two fatty acid molecules, which are surfactants, and acids, right? So you got some protons there. So that's where the pH gradient comes from. This is a very simple chemical reaction, spontaneous reaction that's happening that allows this thing to move, basically. Okay. So just some very light technical details. This droplet is sensitive to the pH of the environment, and it's creating, by the way, its own pH by making acids. And so at the surface of a droplet, you're going to have the self-assembly of these surfactant molecules, and they're going to have two states. They're going to have either a deprotonated state or the protonated state, and that's it. It's like a binary switch. You could have one or the other, or a mix of the two. And by switching the protonated state of this surfactant here, you're basically changing the tension along the interface. Okay, so here's just some readings of what that could be. So if you're starting an experiment over here at pH 11 and you start creating acids, the pHs start to drop, and then suddenly your tension goes way up. Okay, so there is built into the system a small molecule that is sensitive to the pH of the system. And that small molecule, when it switches from ionized state to the protonated state, is changing the tension along this membrane. Okay, that's important to understand. And so a little more on this mechanism. When that happens, you basically have a gradient across the surface of this droplet. And when you have gradients in physics, for example, it's quite common to see some sort of dissipative structures such as convective cells. So if you look at this micrograph here, you'll see these droplets that are slowly convecting. So each of these turns into a convective cell, and then they start moving through the experiment because they're convecting. And so the pH that the droplet produces is responsible for this tension imbalance around the surface. And that's responsible for fluid motion, which are these convective cells that allow these things to then to have some fluid dynamics. And because we're working at low Reynolds numbers, again, it's not important that you understand this, but what's important is that you understand that viscosity dominates. So when this thing convects, the surface of the droplet is moving, and when it's in contact with water, it's creating friction. So these are basically swimmers. When you see them moving, they're swimming through the water phase. It's very similar to how we swim. When we swim, we put our hands out and we create friction with the water to propel ourselves forward. So that's what these droplets are doing, creating friction with the water to propel themselves in a certain direction. So that's what all that technical stuff means. This is the non-technical thing. If you didn't understand any of that, maybe you understand this. This is a common toy, at least in the US. This is like a balloon that's a circle filled with fluid. And if you ever played with one of these, you grab one end of this toy and the fluid displaces when you put tension on one end, and the fluid displaces here and the center of mass moves, and you can't hold it anymore. It just falls out of your hand. So when you put tension on it, the thing moves its fluid in order to get away from you. And when it gets away from you, then the tension is equalized and then the thing stops moving again. So this is very similar to what's happening with these droplets, is they have this tension difference on one side versus the other. And so the droplet tries to equilibrate this by moving its fluid. And when it moves its fluid, it then propels itself through the system. So that's just a little example of this. Another way to think about this is from this physics example here. So in this particular case, what you have is external energy, right? That's being generated. And then you have this agent that's in the right place at the right time with some sort of detail as far as what's the shape of this object here, right? In order to harvest the energy in this particular wave. And so what you're seeing with the droplets that are moving through the system, they are generating their own wave in a sense. It's kind of just it's a chemical wave in this sense. But they're creating their own wave, and so they're surfing on their own wave, and this will occur just like the surfer here, as long as there's energy to push that droplet around. Okay, but the thing about this, again, about this droplet, the wave doesn't come from outside. The wave comes from the droplet creating its own chemical wave as it's traveling. That's shown here. And you could see part of that chemical wave by seeing that pH difference around the droplet. Okay, so that's how that stuff works. I hope that's clear. And so at the end of all this, what we have is an interesting example of chemotaxis. Again, movement in chemical gradients. And what you see on the right is a living natural cell called dictyostelium here that is sensitive to cyclic AMP, small molecule that is being put into the experiment by this pipette tip here. And that organism senses the CAMP, and because it has receptors for it, and then reconfigures its cytoplasm to flow the cytoplasm towards where the source of the chemistry is. So that's biological chemotaxis. It's very sophisticated what's happening in this particular case on the right. But with our little droplets, we're able to do quite a similar thing. As you can see here, we're going to make a road of chemistry here. And the droplets can sense this and then move directionally through those things. So we have, a non-biological example of chemotaxis in this case. Okay. So I talked about these before. We still have these particular properties in the system I just described. We're also going to add to this list sensing. So we have this molecule that's sensitive to pH conditions. We have motion because we have a fluid droplet that can do things like convection. And this droplet is interesting because it couples the sensing and the motion together to make sensory motor coupling, which then allow this thing to perform now this kind of chemical, non-biological chemotaxis for us. So now we've doubled the list of things that we think are interesting to study with these droplet systems. Okay. The sensory motor coupling is interesting. So this is something that's been widely studied in biology as well as far as neuroscience. So how you build up. Once we started animating these droplets, we just wanted to look at motion, basically self-motion. We didn't expect to sort of get this far with the interesting dynamics of these systems. And this kind of reflection upon what does this all mean as far as these non-living things that show sensors and motors coupled together. They can move directionally in gradients. What they're effectively doing by doing this is, this acid trail that you see here, this gradient, is a waste product of the system. So what these things are actually doing are basically using up the fuel, producing waste, and then moving away from the waste products. And from a chemical point of view, what that means is that this is an emergent mechanism to avoid equilibrium in these things. If these droplets were encased in their own waste products, they would be at equilibrium sooner. But the fact that they have an emergent mechanism to move away from their chemical waste products, that means that they're sort of sustaining this non-equilibrium state, which I think is interesting and may be very similar to what we are doing in biology, having mechanisms, and sometimes very sophisticated mechanisms, to avoid an equilibrium state. So is this an example of self-preservation? We go as far as saying, in this simple chemical droplet, is it telling us something interesting about self-preservation that we thought maybe would be reserved for living systems, certainly. But what about these non-living systems? Are they doing the same thing? That's an open question for you. Okay. Then just for fun, and I won't have time to get into this, but if you put two active droplets together, they both have the same chemistry. They just have two different dyes just for a color effect. So you can see the result here that it appears that the action of one droplet is influencing the action of the other droplet. And this is statistically significant and quantifiable. And I don't have time to get into the results here, but I just want to show you this, that when you increase the number of droplets from one to more than one, things get very interesting. So can we, for example, put dancing in our list of fundamental properties? I'm not sure about dancing, but at least group dynamics we can put in there as another interesting property of these systems that we're exploring. Okay. So here's an even simpler system. This droplet is in one of these dishes and is floating on the surface, and it does not contain any fuel. Okay, so it's just a droplet that's passive and does not contain any fuel. So it's even simpler than the one I just showed you. So it's the surfer again. It's more like the surfer this time in that the droplet doesn't do anything on its own. It'll just sit there in the water until the wave comes. When the wave comes, if it could catch the wave, then this droplet can move, but it's not creating its own wave anymore. Okay, so it's a passive droplet, no fuel. So here's the droplet put into the system, and it's just going to kind of float around waiting for the wave. The wave is going to be put in here. You'll see a pipette come in. Okay, there's the pipette. It made a chemical gradient. The chemical gradient is slowly diffusing across this chamber here. And once the droplet can sense the gradient, because it can still sense these gradients, then it'll catch the wave and it'll start moving. There it goes. Okay, so you can get really simple chemotaxis even without having any fuel in these systems. So you can do something fun like this, where you put your droplet here. It's not motile yet. Here's the source of chemistry of the chemoattractant here. And then the question is, can that droplet find the source of chemistry in this maze? Yes, of course I can do it, right? Of course, that's why I'm showing you this example, because it works. And the issue is that, remember that these droplets, when they're sensitive to the environment, such that the tension is changing from one side of the droplet to the other, you're basically putting some sort of pressure, let's say, on one side of the droplet. So the droplet then needs to move to try to equilibrate that pressure. So it's basically trying to find the lowest energy state of the system. So what is happening here with this particular example is, even though the droplet is initially here, this is a homogeneous environment until we put in the source of chemoattractant here. And now there's a kind of chemical wave being dissipated through the system. And so that's when this thing is sensitive to it. When it picks it up, it's able then to move directionally towards the source of this. And the fact that it looks like a maze to us doesn't matter, because all it's doing is energy minimization in the end. So it's just a very simple process. Okay. So those are some interesting properties of these living systems I want to talk about just briefly today. And now I just want to give you two examples based on these self-moving droplets. So one is a potential application, the other one is an origin of life scenario. So we'll start with the potential application of this kind of stuff. So here's another video I'm going to show you in a second. It has two droplets in it. And these two droplets are the same kind of droplets that you just saw in the previous movies. So they're sensitive to the environment, and they can follow chemical signals and things like this. And so they're sitting around, and these droplets have been modified a little bit. As you can see in this inset here down here, that there are these little things sticking out in the side. So these are little packages of alginate. And in these packages of alginate, we can put stuff in there. And the idea is, well, can we use these, since we can control some of the motion of these droplets, can we use them as transporters? So that can they move something from one place to the next and then deposit it in a certain place and then go away, just like a delivery system. So if you order something from Amazon, they're going to package it in a box. So these are the boxes here. These red droplets are the drivers that are going to bring the boxes to your house. When they get to your house, they're going to drop the boxes here, and then they're going to go away. So that's the same kind of thing that's happening here. So here's the chemical attractant being added to this part of the system. Of course, these are sensitive to that, so they move to find the chemical gradient. Now there's a decoupler, so it's saying, okay, please leave the packages here, and now move out of the way, go back over here. Okay, great. So we're able to do something fun with this as an example of these kinds of things. And we're able then to, what can you put in these packages? Well, you can put cells on the packages. So we put some sort of bacteria and stuff in them. These are the packages. You can see these little white circles. And then after transport, we harvest those packages and we say, are these cells still alive? And in many cases, yes, they're still alive. It's not trivial to say that because this chemical experiment is at very high pH. You see pH 11.5. It has some, let's say, nasty chemicals in it, like decanol and things that cells typically don't like. So in order to sort of mitigate the sort of, it's actually a sterile system. If you put cells directly in these chemical systems, they die. So it's a sterile system. To mitigate that, we made these little capsules here, which are alginate, so they're hydrogels, and then we can use that to protect the cargo, just like you would put your glasses from Amazon in a box. So when they transport those glasses to your house, it's in a box and they're protected. So that's what the alginate's for, and that's what these little round things are. These are the little alginate boxes that we put the cells in. Okay. And then finally, with this example is, okay, well, that's bacteria. They're kind of robust. What about human cells? And so we tried different kinds of human cell lines to transport in the system. So you grow up some cells here, you encapsulate it in the package, you incubate those packages in some growth buffer, and then you put the package with the droplet, you put the droplet in the arena, you put a chemical signal, and then if everything goes well, the droplet will transport the cargo from point A to point B. You harvest these packages, grow the cells, are they still alive? Those kinds of experiments. So first, what's interesting is what we had done here is taken some human lung cells, which have been known to produce biosurfactants. And so that's what's indicated by these little sticks here, these black sticks in the system. So if these cells are doing fine inside of these packages, they're going to produce some of these surfactants, which could then coat the package. The package gets inserted in manually. We put this manually inside of this droplet, which also has, again, these surfactants on the surface. So what we then don't do in this experiment is we don't add for example, the pH sensor or the surfactant to the system. So what I explained here earlier is this whole system works because you have a small molecule that then is self-organized on the surface of the droplet, making it sensitive to the environment so it can respond. Well, we don't do that anymore. We don't add this molecule to the system. We let the cells make these molecules and add them to the system. And if the cells don't make these molecules, well, that package does not get taken up by the droplet. So that doesn't work. And secondly, if you want to transport this, you need those surfactant molecules made by the living cells to act as the sensor in order for the droplet to move from point A to point B. So that is also, again, important for what the actual contribution of the cells are to the system. And this only works as a transport system if the cells are alive. So what we say from this is auto-selective transport. This is selecting for cells that are alive under these conditions. And so this could be expanded to other things, but basically what we're saying is there's something about the products produced by this cell that then allow for the whole system to happen. So you have a fluidic transport selectively for living cells or for living cells that are producing the molecules that you want. So that's why it could be considered maybe a future application of these types of systems. Okay. Lastly, I want to speak about an origin of life scenario, which is one of the reasons I got into this in the first place, because people thinking about origin of life tend to like to think about simple systems that can do cool and interesting things. And so I think at least you get my perspective so far that these droplets can do some cool and interesting things. So can they do anything that teaches us something about an origin of life of some kind? And so I like to start this kind of argument by talking about the origin of life, a perspective on the origin of life. As you guys know, when you work in the laboratory, you need very sort of clean conditions, orderly conditions. And you buy your reagents from Sigma Aldrich or someplace like this, which basically gives you 99.9% pure stuff. And you have to be very careful on how you control the initial conditions for your experiment, or the experiment doesn't work anymore. It's very unlikely that you had such a very well-controlled biology or chemistry lab at the origin of life. All the chemistry that we know that is self-generative, that, for example, can produce chemical bonds, is really uncontrolled chemistry. You get combinatorial explosions. You get huge suite of organic molecules. So what that might look like is shown in this picture here. On the right, you have a vial of a 99.9% pure sample that you can buy from a chemical supply company. And on the left is what happens when you have an out of control chemical reaction. This happens, when you cooking up sugar in your kitchen and you leave the heat on too long, it goes from a nice white powder into some black stuff that's hard to clean off, right? So this is the chemistry this is likely the chemistry that the origin of life had to deal with in some way in order to make some sense of this stuff. So a huge amount of chemical complexity, let's say. There's also, you can look beyond our planet out into what we know about our solar system, and there's some interesting chemistry going on one of the moons of Saturn called Titan. It has a dense atmosphere, and it appears to have stable bodies of surface liquid. So you can see even the reflection of light from the surface of this moon. These are basically liquid. This is liquid methane that is on the surface, right? Not liquid water, right? So there are other kinds, I guess, when you're thinking about origin of life in a sort of broad perspective, there are other kinds of chemistry that one may want to consider when doing a chemical experiment, especially a messy chemical experiment like I'm going to describe. And and Titan has a very active chemistry as far as we know. For example, a lot of the chemistry is based on hydrogen cyanide, and hydrogen cyanide is a very simple molecule as shown here. But this thing could self-polymerize into larger and larger and more complex organic molecules. This has been known for a long time. And so the idea here is, well, this is an active chemistry that's likely happening even in our solar system. Can we use this kind of chemistry as an example of prebiotic chemistry that could give rise to something interesting? So what that means is we took this hydrogen cyanide here, and instead of doing one of these self-moving droplet experiments with pure anhydride like I showed before, which is this pH sensor and all that stuff, take that out, and we do hydrogen cyanide polymers and throw that into our system as a potential source of fuel. So we don't give it pure fuel anymore, we give it this stuff, whatever this stuff is. And what I mean by stuff is, I mean stuff, because when you analyze this, for example, with mass spec, people estimate there are over 50,000 different types of organic molecules in that stuff. So it's really a big mess is what it is. So we made this polymerized hydrogen cyanide in the lab. This is us making it. There's a trap here to try to avoid, hydrogen cyanide, it sounds bad, right? And it is bad. It's a poison. It's terrible. So we were doing this in a proper chemistry lab with the right safety controls. And you really needed to do that because you're producing potentially dangerous hydrogen cyanide. But once you make the hydrogen cyanide, it's interesting that it then reacts with itself. So it self-polymerizes to making more and more chemical bonds. And when it's polymerized, it's no longer harmful. It's just when it's in a monomeric form, let's say, that it's harmful. So here's me in the lab holding up some of the experiments. So this is early on in the experiment, this is midway, and this is later. And you can see the color change of these samples as the hydrogen cyanide is polymerizing, making more and more complex organic molecules here. And it's at this stage that we harvested these molecules, whatever they are, and used this in a droplet experiment. So here's some movies based on that with a microscopic image. And again, instead of putting in the fuel, we took out the fuel and put in this stuff. And what I mean by stuff is this black stuff. You see these black particles. They're black particles in the oil phase, in the oil droplet itself. They're black particles on the surface of the oil droplet. They're black particles in the water phase. I mean, it's just a really big, messy experiment. And so we then see the dynamics of this. So these are fresh droplets put into the system. And you're watching the first few seconds of this. And already you can see the dynamics. You get convective motion emerging from these kinds of systems, again, without the chemical fuel that we saw before, only with the HCN stuff. And can it do chemotaxis? Well, here are some convective HCN droplets here. Here's some chemistry I put in here. And yes, they're also responsive to chemoattractants. So they're not only motile, but they also can sense the environment and move throughout the environment like we saw before. So what I think is interesting about this is we can make this very complex chemistry that appears to be available in the universe that then can support self-movement of these systems and also chemotaxis of these systems. And what I find very interesting in this is the fuel for this is not explicitly defined. And what that means is, you know, when you do your experiments in the lab and you make a mistake in the initial conditions and the experiment doesn't work, you'd be very careful, right, about what exactly you're putting into your experiment, or it could not work. Well, when we did this experiment, we made this HCN polymer and we didn't purify anything. We just took what was ever in that black tube and mixed it up with our oil droplets and did the experiment. So we didn't do any purifications. There's no fractioning of any of these products. They're all in there. And so the droplet kind of has to sort itself out. If it's going to move, it has to figure out how to sort out its own chemistry in order to actually move in the system. And so they think this is interesting because the oil droplet is then a simple structure, self-assembled structure, powered by this potentially prebiotic chemistry that is interesting because the structure itself is avoiding equilibrium, again, like living systems do. Okay, so that's the end of the content for the talk. Let me just go through a few conclusions before we get to any questions. So what I wanted to show you today is using these droplets to do some fun and interesting things. When you couple chemistry with droplets, you can produce self-moving systems and some kind of artificial chemical chemotaxis, and you can make them do funny things like solve mazes. You could also use them for stuff like transporting cells around. My collaborator calls it a chemotaxi for these experiments. It could also be selective for living cells and their products. And also messy origin of life conditions could also power these kinds of systems. They don't do it so well. I mean, the movement is not as great as when you use purified components, but the system still works in principle. And so what we're interested in here is how these simple, and you know, apart from the messy chemistry stuff, these are simple chemical systems that we're finding are intimately coupled with the environment. That's key, that these things could act on and react with the environment. And that's why it makes them so interesting. And so what you see are these emergent mechanisms to avoid equilibrium. And what we're playing with now is the sort of interface between our artificial systems, so our droplet systems, and living systems, and how they complement each other to produce a functional coherent system like this auto-selective transport system. And then finally, even highly complex, messy systems that may be more interesting and relevant to an origin of life scenario can self-organize equilibrium avoidance and perhaps even, question mark, self-preservation. So this is what I want to talk about today. These are some of the sort of properties we've explored and discovered with these systems. There are a lot of things about living systems that are not on this list. So there's quite a lot more work to be done. For example, where is evolution on this list, right? That should be another part of the discussion. And so I'd like to thank the organizers again for inviting me. And I also like to thank funding agencies and the projects that gave rise to this work. And thank you for your attention.
Okay. Okay. Thank you very much. I'm sorry, just because otherwise we have the echo. Thank you, Professor. Now we are here for your questions. So don't be shy. You can write them in the chat. You can open your microphone and just ask. Also, if you feel more comfortable, you can ask them in Italian. And so, okay. What about the little jet that the dirty droplets left when they were moving? And have you ever tried these moving particles with a double emulsion architecture?
Okay, cool question. Thanks. Oh, that's Aitor. Hey, thank you, Aitor, for the question. Okay, what about that little jet, the dirty droplets? Yes, when they were moving. So, in the dirty droplet experiment, Aitor was quick to see that that the little particles of hydrogen cyanide polymer, some of it were ejected out the back. And what I didn't mention before is that when we do the work with the anhydrides and the fatty acids, those fatty acids also get injected out the back. So so part of the motion that we see from a physics point of view is basically we're talking about rockets, right? You're pushing mass out the back to move forward. That's what happens when you have a lot of chemical energy in the system in the initial stages. That then wears out, and then that's when we see these sort of fluid dynamics take over with the viscous drag on the system. So the movement of the droplets is a bit more complicated than what I explained, but the idea is, yeah, you have different modes of motion, and some of it has to do with the just laws of motion, that you just lose some mass out one way to move the other way. So that's what's happening there. Have you ever tried these moving protocells with a double emulsion architecture? Mm-hmm. I've made double emulsion architectures before, but I don't remember doing any kind of motion with those. The motion, now that we're getting into it a little bit more, if you want to get these systems to move, even if you want to get, for example, a vesicle to move, you have to act on it, right? If the vesicle is going to move on its own, I think what it has to do is it has to lose some mass out one side in order to move in the other way. And if you're not going to do that, you need to do a convective cell. And so if you could do a convective cell, the reason I'm saying this is you have a double emulsion architecture. I don't see how you're going to preserve double emulsion architecture with a convective cell, but that's something we can discuss.
All right, thank you.
Hello, Professor. So I have a question, if I may.
I don't hear her, do you?
Do you hear me?
I don't hear her on my computer. Yeah.
Okay, if you can just ask again.
Okay, so sorry. My question is about this moon of which you talked before. Did you already check which type of molecules may come from these polyimine structures? So maybe if I don't know, amino acids or something similar were formed, so like something at the origin of biological life. I don't know.
Yes. Thank you for your question. Sorry, I have a little echo here. We didn't check because these systems are so complicated, but other people have checked with the same type of hydrogen cyanide based system where you can get different kinds of functional groups like amine groups and carboxylic acid groups. And the HCN chemistry has now been shown to make many different types of interesting biological molecules, such as amino acids, like basically your question. Whether there are amino acids in the experiments that we did, we don't know. We didn't check for them. But what I could say is we did check for, for this system to work, you need to have these tensio-responsive elements. So you need some molecules that are going to change the surface tension of a droplet. That's the only thing you need. You need something that's going to change that tension along that interface. And so what we did then test for are these kinds of molecules. So there are molecules in that mix of stuff that are very sensitive to pH, and those are probably the molecules that are then allowing these droplets to convect and to move around. Yeah, but yeah, other people have shown very interesting results with the same chemistry, making amino acids and things like that. Very interesting stuff. Yeah.
Thank you.
You're welcome. Any more questions? I have a question here from Richard. Hello, Richard. Do you have any ideas of how to introduce evolution in a simple system? That's good. Thank you, Richard, for that one. Yeah, how would you introduce evolution in these systems? No, don't be sorry. Don't be sorry. So I'll try to tackle your first question first. Yes, how to introduce evolution in the system. So we have, and I didn't present it today, but we have made a rather interesting cycle of fission and fusion with these droplets. So we have droplets that will self-divide by basically changing the environment that they're in. They'll just break apart into smaller droplets. You can transfer those manually. I transfer them manually to another tube where you have other, let's say, food droplets, and then they fuse with those so they can make these hybrid droplets. And then I divide those. And then, so we can go through several iterative cycles of fission and fusion, which I think is not yet evolution, but forms what we need to do. Then what we need to do is basically add some diversity to the system and have that diversity somehow govern these fission and fusion properties, which also works, by the way. And so that's something that we can discuss. And therefore, the thing that's evolving is basically the chemical composition of the droplet and the size of the droplet. I've also seen that seems to be important for these kinds of systems. So yes, in principle, then we can start to get evolution going. Now, whether this suffices as Darwinian evolution is debatable, but it is a kind of evolution at least. Second question is, would it be possible to introduce some kind of competitive chemistry? Yes, it is possible. It's very possible. What we've done so far is very little in exploring how chemical reactions are going to influence the behavior of these systems. I talked about one, which is the hydrolysis of an anhydride, and this stuff has been studied for a very, very long time. With Louise's group, for example, they did a lot of work with this and others. And so that's it, right? And so now there's the whole world of chemistry out there that in principle could be useful for us to increase the complexity of the chemistry, but also then to, as you're sort of asking about, introduce competitive pathways in the chemistry. I think that'd be very interesting.
Okay, can you hear me?
Yes.
What I mean with that is can the competition in of itself be the beginning of an evolution, right? Because that way you have a sort of selective pressure, right? Mm-hmm. If you have, so there's different systems competing for resources.
Yeah. Yep, that's true. So yeah, thank you for clarifying that, Richard. So there is an interesting how should I say? There's an interesting. Give me a moment to think about how to say this. Because what you had said now is, in your question, is you need to have a competition for resources. Okay. So that means some of the chemistry that's involved in this needs to come from the environment, and then the droplets, I think, need to go and find the resources in that environment, which they can do. There's an interesting paper from Sugawara in Toyota in Tokyo, where what they've done is they've changed. They have a droplet system that's moving around, and they've done something very sophisticated and different than what I've shown. Is they have the catalyst is in the oil droplet, and the resources are in the environment. So they have to move. They move through the environment, collecting resources, and then they get catalyzed by the droplet. And so I think that kind of thing, it's like how do you embody these systems is very, very important. And that kind of system, for example, in that kind of system where the droplet holds the catalyst, then the droplets are, in a sense, immortal. As long as you keep feeding them, they keep moving, right? My droplets don't do that. My droplets have a certain amount of fuel, and they eat it up, and then it's done. And so, yeah, I think therefore you could have different kinds of droplets in the same environment competing for resources. The droplets that are successful in getting the resources, let's say, are growing or maybe dividing, depending on how you want to do the chemistry. And then you try to do several generations of this. I would guess that's how you would do it.
Very cool. Thank you.
I'm
sure we'll discuss this more.
And we will. Please come to Trento.
Of course.
Any more questions?
Okay, okay.
Okay. Do you hear me now? Okay. Thank you, Francesco. Do you think reproduction is a fundamental biological property, or living things just have to be self-sustaining chemical systems? Well, you want to know what I think about this? It's a big question for me. I think most people think reproduction is fundamental, and it seems like the kind of biology that we know, which is life on this planet, reproduction is, yeah, it's essential, right? However, I think there's some credibility to this contrary idea that living things just have to be self-sustaining chemical systems. What is the NASA definition is a self-sustaining chemical system capable of Darwinian evolution, right? So it depends on how you interpret that. But that's very heavy on self-sustaining chemical system, the NASA definition. So, but is capable of Darwinian evolution, I think implies reproduction. That part of the definition, I think that's where that's going. And one of the issues, one of the problems we have is that we only have basically one biological plan by which we're trying to learn everything. So biology always follows the same central plan, right? We only have one example of living systems. And we don't have an example of a completely different living system to study. So our thinking is biased. We are biased because of the kind of data we have available to us. So I would like to think that perhaps there are other kinds of systems that are, let's say, viable. And one of the fun discussions we had in my group a few months ago is a question that I posed to the group. I said, why do we need to reproduce? Why don't we just get bigger? that would be fine. So you get resources and you just get bigger. And the bigger you get, the more competitive you are, because you basically, leave no space for your competitors and just keep getting bigger and bigger. And of course, there are physical limitations. The biggest single cell organisms are ones that live in water because of gravity, right? Because if you're living on land and you just keep getting bigger and bigger, then you're going to suffer from gravity. So if you can float around in water, which we can do on this planet, then you could just keep getting bigger and bigger, perhaps. And then I think the interesting argument against that is, well, in order for living systems to work, you need to worry about diffusion limits and things like that. So just to have signals and molecules pass from one side of the cell to the other is probably important. So you shouldn't get too big. And maybe cell size is the right size for diffusion systems. But again, without having very good counter examples for us to study, I don't know, I think we're probably just too biased by this. But it's a very good question. Thank you, Francesco.
I have a question. Can you hear me? Can you hear me? Can you hear me?
Yes, we heard you.
Okay. You mentioned before in Tokyo that they did an experiment with an catalysis molecule inside a droplet. And could it be defined as alive? I mean, what's your definition of an alive system? What do you think? What are the characteristics, the basal characteristics for a system to be defined as alive?
Thank you. Can you hear me now? Is it coming? Yeah, good. It's a little delay. Thank you. That's a very good question. So the example I gave of the catalyst and the droplet seeking resources, I wouldn't claim that any of these are necessarily living systems that we've talked about today. I did transport living cells, so probably those living cells are living systems. But again, I'm not quite sure. But I don't have a definition of a living system. But if you ask me what's important for these, are some of the properties listed that I listed in that slide. I think those are particularly relevant for primitive systems, for example. Maybe origin of life scenarios, very, very important. But what is not on that list, and what I would say is very, very important, is evolution of some kind. We're missing evolution so far, and that is, if you're able to show me one of these kinds of self-sustaining systems that avoids equilibrium and also has the potential to evolve, I would say, well, we're getting pretty close to what life is there. It becomes a philosophical but also a political discussion, right? Beyond that, because that's a big that could become very controversial. But that's what I would say. I would like to see one of these self-sustaining chemical systems that is indeed capable of evolution. And now I'm not so biased towards Darwinian evolution. I don't know if it needs to have a genome. Not sure about that. Probably needs to have information, probably needs to have heritability. Maybe it could be Lamarckian evolution. In fact, I would be pretty satisfied with Lamarckian evolution. That's my answer. It's not a very good answer. It's not a very scientific answer either. It's kind of like more of a pub discussion answer.
Okay, thank you very much.
You're welcome. Any more questions?
Okay, I have a question. Do you think that exploration of other planets, maybe Mars or the Moon, could lead to discover other interesting aspects regarding the periodic chemistry? Yeah,
I think that could be, if we're able to find evidence of of life on other planets, for example, Mars, which of course people are looking for actively, right? So, and a lot of money is going into that particular question. That is, I think, reasonable because perhaps the conditions that allowed life to flourish on this planet were similar to the conditions originally on Mars. This is part of the thinking. So that's hopeful, and maybe that'll give us a related ancestral sort of data point that it could be very different, hopefully very different from life on this planet. However, there are caveats to that because if an exploration of space in our solar system will allow us to come across a different form of life, we have to know what we're looking for, in fact. And I think, by and large, what we are looking for is water-based life. So this brings up a point that I didn't talk about in my talk, is if we're trying to understand something about living systems by looking at oil droplets, well, that's kind of cute and all, but you know, living systems are not living cells are not oil droplets. This is a really weird way of looking at it. We are, again, biased by our biology, and we spend a lot of money looking for signs of life that are more or less like our own, you know, and that's reasonable if you think that's what you want to find. But then we have to really expand our perspective to try to understand what other types of life are possible. For example, there's some speculation that, you know, you need to be liquid to be alive. This is an assumption, and could be debated. But let's say you need to be liquid to be alive. Well, there are many different types of liquid, and you could run a lot of very interesting chemical systems without water. You know, you do a lot of organic chemistry, and usually when you do organic synthesis or in organic chemistry, you do this without water because water is a problem when you want to make molecules. In fact, this is another kind of conundrum for these origin of life scenarios, is if you want to make something like DNA and you want to make it from scratch in water, good luck making those bonds, because water is going to just hydrolyze whatever you're trying to form as far as carbon-carbon bonds and things. So water is the solution and water is the problem for a lot of these things. So the more information we can gather on what's happening or what has happened on other planets and other moons, I think that's going to be interesting. So there is also a lot of effort to analyze these different types of moons, because we're finding they're really interesting states and atmospheres and compositions on these things, and that could give us a little bit of a wider perspective, I think, on what is possible as far as life. We only know about life on this planet so far, and we know about water found in different places. We know about organic compounds found in different places in the solar system, but I think that's as far as we got so far. There's a question here from Enrico. What is the role of robotic interfaces in your research? Well, we've done a few things with robots. I didn't mention them today. It has to do with the fundamental principle of these things is working with systems that are far from equilibrium. I think the reason that these systems are fascinating is because we push them far away from equilibrium, and then we see self-movement and all these other things I talked about. And so one reason is we can use a robotic workstation to interact with our chemical systems in real time and keep them away from equilibrium, like, from some sort of external interference. So that's one thing. And the second thing is, what we'd like to do is see how these robotic interfaces could drive maybe some of these concepts we talked about in the discussion today. For example, can these systems be selective? So can you use a robotic arm, for example, to select droplets in a population and use that to drive evolution eventually in these systems? So that's why we've worked in the past with robotic workstations specifically designed for working with our oil droplet experiments. You're welcome.
So, do you have other questions? Or if there's no one else that has something to ask, I think that we can go towards an end. Okay. Well, so thank you everyone for being here today, and thank you, Professor. It was really nice to listen to this research. So just to remind you, you'll find this conference online on YouTube. So if you lost, I don't know, like the start at the end or something, and this was Co.Scienza Festival, and you will have other events tomorrow and on Wednesday. So follow us on Instagram or just look at our website. And that's all. So again, thank you very much. And we'll see you soon, I hope so. And thank you, Professor.
Thank you.